Now let's go on to our discussion of the coccidian parasites. So let's get going and tell you a list of all the coccidian parasites that we have. We have Toxoplasma gondii, then we have the three which belong to a similar family, that is Cryptosporidium, Cyclospora and Isospora. And then we have something which you really don't have to focus on much, and that is going to be Sarcocystis. So let us start our story first with Toxoplasma gondii. As soon as I say Toxoplasma, what should be coming to your mind is going to be a cat. It is very, very important when you think of a cat, because the definitive host over here is the cat. So this is going to be one part of the life cycle that will help you identify that the cat is definitely there, and the intermediate host is then going to be us, right? Of course, this has involvement of a lot of other animals also. This can be involving the sheep or the rodents or the pig. Other animals are also there, but definitive host to the cat means that the oocyst production, the sexual reproduction, that is what is the definition of a definitive host. Now, definitive host is the one in whom the sexual reproduction is going to occur, or the oocyst production shall occur. Now, I'll come to oocyst, but I want to tell you that what are the infective forms. You'll say till now, everywhere you had told us that trophozoite and cyst, these are the kind of forms you were talking about. Suddenly, I'm talking about sporulated oocysts, tachyzoids, bradyzoids. Why? Because I've entered into the sporozoa family. That is why now the terminology has changed. So one of them should have been the infective form. What is this? That is exactly, Toxoplasma is really toxic. Every form is infective. Sporulated oocyst can also cause infection to me. Tachyzoid can also cause infection. Bradyzoid can also cause infection. So now, how will I get the infection? Because as I said, sporulated oocyst can come, bradyzoid can come, and tachyzoid can come to us. So please note, sporulated oocyst is going to come through soil, food, water, all contaminated things. So if I end up consuming any contaminated food and water, the sporulated oocyst form will come to me. Okay, if on the other hand, now look at these two things. I'm going to make a story of BT-BT. What is BT-BT? Means if I am having a patient who has received some faulty blood transfusion, one is blood, say, so blood may everything will come. Blood will also have blood transfusions, blood will also have, what if it's a mother to child? Mother to child also blood only goes, na? So mother to child wala history bhi hoga. That is one scenario. The other scenario is coming from some tissues. How will I consume tissue? Some animal's tissue means I'm having meat. I'm having some undercooked meat. So now there are two situations that I've put up in front of you. Now you just have to make the mnemonic BT-BT. Means if they have written the B, that there is an element of blood transfusion, you will put the T, which is the form that is going to enter into the body. You will say it is tachyzoids. And if they have said that there is something to do with T for tissues, then you are going to say, okay, then I'll write the B. This means the bradyzoids are going to come out of it. Is that okay with everyone? Easy to learn, na? Sporulated oocyst is something which is going to be coming from contaminated food, water, soil. The bradyzoids will come from some tissue, and the tachyzoids will come from blood. So BT-BT wala mnemonic rakhenge. One more thing, out of bradyzoids and tachyzoids, which are the ones which are actively multiplying and which are the ones which are slow multiplying? So think of your bradycardia and tachycardia. Convert it into that, na, bradycardia, tachycardia. So tachycardia matlab heart rate going up, bradycardia matlab heart rate going down. So tachy means jaldi. This means this is actively multiplying. This is the form which is going to show you active multiplication. And then which is the one which is going to be slow and dormant, and that is bradycardia matlab bradyzoid. So simple. Cardia wale se humne yaad kar liya. Now I can show you the final life cycle. So this is the life cycle where we are to gonna come very late. The actual story is happening a lot between animals. And you know this cute little cat. As soon as you will see a cute little cat, you need to know that we are talking something to do with Toxoplasma gondii. This is something to do with Toxoplasma gondii that we have. So what do we have? What is this cat going to do? Although she's very cute, but we're going to make her shit. So basically she's going to shit out the organism in her feces. So what is it? She has sent out oocysts, because definitive host, na? Cat is the definitive host. So definitive host mein kya hoga? The sexual reproduction will occur. If gametocyte, sexual reproduction will occur, this means the oocyst will form. You say, is the oocyst, now I don't know how many of you notice this. Is oocyst infective? Let me ask you, if I tell you to select what is the infective form, and you have two options in front of you, oocyst I know many of you will select oocyst, but if I say the other option is sporulated oocyst, what is the infective form? You will say, oh, okay, ma'am, only oocyst is not going to be the infective form. No, no, no. It is the sporulated oocyst. So this means when the feces are fresh, so this is so sad now that not only this cute little cat, I'm discussing her shit, and then after that I'm discussing whether the shit is fresh or the shit is old. So when her feces are fresh, when the feces from the cat are fresh, they are just oocysts. It is only that when we are gonna have certain old feces, and that is the time that I'm giving it for it to become sporulated. So if you get a question in which you have in your previous year, is there gonna be infection with fresh cat feces? No. There is gonna be infection to the humans when there is contamination with old cat feces, because by that time the oocyst has sporulated. So this was the method of coming to humans. But I told you, na, human to baad mein hai. Actually, it is going on and on between animals. So imagine if this is a cat, she has oocyst. So you'll say if the cat is eaten up by other animals like a pig or a rodent or a sheep, so if it is going to be eaten, now if someone asks you that, acha, in sub mein, which is the form that is going to be in the blood and which is the form that will be sitting in their muscle and their tissue, you'll say very, very simple, that BT I will do. If you ask me which is the form in their blood, I will tell you tachyzoid. If you ask me which is the form in their tissue, I will tell you bradyzoid. I know how to do BT. So one way was that via this particular cat and her feces, the sporulated oocyst came to us by contaminated soil water. If it comes to us via eating undercooked meat, maybe undercooked pork or any other undercooked animal that you eat, so tissue ke through kaun sa form will come to us? Tissue ke through bradyzoid will come to us. And if this is coming via blood transfusion, either it is blood transfusion or you can say mother to child, pregnancy mein, then what is it going to be? Which form? So blood transfusion mein kya form aayega? Tachyzoid aayega, BT. ठीक है. Now let me see how many of you were listening to me carefully and how much of application can you do. If I ask you, you have a patient who has undergone organ transplant. This is gonna be one of your not extra age pata hai, but application is important na. When I'm talking about organ transplantation, someone got toxoplasma via an organ. Organ matlab what? Organ matlab tissue. Tissue matlab what is the form that has come? It is the bradyzoid that has come, hai na? So, and the concept, see, I told you mnemonic is still the same, but they've twisted it that someone got organ transplant done, tissue aaya matlab bradyzoid aaya, BT. So this is done. Now I can move on to that. In all of this story, I had also mentioned mother to child. This means this can cause congenital toxoplasmosis. Let us start off from there. I'll talk about a congenital situation. I'll talk about an adult situation. I will talk about an HIV wala situation. So let's start off with the congenital toxoplasmosis. When does the most severe infection occur? If the infection gets transmitted in first trimester, because everyone knows that is the time of organogenesis. However, most common transfer happens when? For every infection, the most commonly happens in trimester 3, because especially during the vaginal delivery, maximum chances of transfer there. But most severe infection occurs with trimester 1. What are the conditions that these patients are going to have? Chorioretinitis. So definitely something very important in ophthalmology that you have. These patients can have, look at this very well, they have cerebral calcification. And they can also have convulsions. I'm seeing this 3 C's. Other than that, there can be microcephaly. Not only microcephaly, they can even have hydrocephalus. So if you read some textbooks, there is always a discrepancy that which is the head finding which is more common. Both of them are there. And of course, mental retardation. A problem है कि जो हमारा cytomegalovirus है ना, which you studied in virology, that also shows you a congenital CMV syndrome, and that is very, very similar to Toxoplasma syndrome. That was also the recent NEET and FMG question. There's a very simple way of learning it. I'll tell you how. And if you go to the virology sections, you shall find. I have told you a way of writing cytomegalovirus. I told you that make a round face, write the C like this. This is the C, this is the M, and this is the V. ये हो गया cytomegalovirus. Repeating, we've written the C, then we've written the M, and then we've written the V. So what are the things that are common between cytomegalovirus and Toxoplasma? Let me see. With the C, I realize that there is C से हो सकता है chorioretinitis, जो कि यहाँ भी है. Then I can see with the M there is microcephalus. There is microcephaly, which I can see over here also with hydrocephalus and mental retardation. That also I can see over here. But what I see in cytomegalovirus is there is a V that is present. Cytomegalovirus ka V, because that is going to show you periventricular calcification, right? That is going to show you periventricular calcification, whereas toxoplasma shows you the presence of parenchymal calcification, cerebral calcification. Remember this very, very carefully. That is why I've told you, cytomegalovirus, the way you wrote cyto then megalo, to kuch features to common the. But that virus finally told me that it shows you periventricular calcification in the brain, whereas toxoplasma shows you cerebral calcification and convulsion. Simple for everyone. This was toxoplasma congenital infection. What about it happens in an adult? Adult matlab fit and fine, immunocompetent. When I'm talking about an adult, I'm trying to talk about an adult with a perfect immunity, that is immunocompetent, and that is going to, our immunity will fight off, na? So mostly it is asymptomatic only. If at all something has to happen, there will be painless cervical lymphadenopathy, if at all something has to happen. Otherwise, it is usually asymptomatic. Also, please let me tell you over here that these patients sometimes can show you infectious mononucleosis like features. If you go back to where did you study infectious mononucleosis? Epstein-Barr virus, na? So Epstein-Barr virus like features can be seen, but that's a rare thing to know. Even if like you don't remember that information, chalega. Okay, now this was if your person was fit and fine, immunocompetent. Now I'll tell you a situation in which your patient is immunocompromised. Now I've got two things. Either the patient is immunocompromised because, our most famous thing, HIV positive, or the patient has undergone a bone marrow transplant, some body transplant has occurred, then also patient is always immunocompromised. So ek ek karke padhte hai, because brain mein they ask you a lot of questions, that when there is involvement of the brain, these patients tend to show you features of what? They tend to show you features of encephalitis. And they then ask you that which is the most common part of the brain that is affected, and that is going to be the brainstem. Two very famous questions. What is the manifestation of toxoplasma in an HIV positive patient? Question 1, encephalitis. Which part of the brain affected? Brainstem affected, and of course you are going to do the CSF analysis. Meningitis, encephalitis hoga to CSF karoge, theek hai? But when you're dealing with a bone marrow transplant patient, usually these come to you with features of pneumonia. And pneumonia mein, when you do a chest x-ray, you will see bilateral ground glass opacities. When you will do a chest x-ray, you will see a bilateral ground glass opacities, and please GGOs, ground glass opacities, and this might get confused with pneumocystis jirovecii, because immunocompromised situation mein pneumonia hona showing you bilateral ground glass opacities can be seen with pneumocystis. So how do I confirm whether I'm dealing with a CSF sample or I'm dealing with a a bronchoalveolar lavage, a sputum sample? How am I going to confirm? I'm going to confirm with the help of a Giemsa stain, which will show me tachyzoids. Please remember, I will see tachyzoids in toxoplasma. That is going to be my confirmation whether I'm dealing with a CSF sample or whether I'm dealing with a bronchoalveolar lavage. So now I've done three things. Congenital mein 3 C's and 2 M's. When I did immunocompetent patient, just lymphadenopathy, nothing to worry. And immunocompromised mein HIV situation alag and bone marrow transplant situation alag. But now, what is going to be the serology? Means antigen antibody wala kya story rahega? Either I'm dealing with an acute infection of toxoplasma or I'm dealing with a chronic infection. So please note that IgM, these are all going to be, IgM is always indicative of acute infection. It rises very quickly. Within 1 to 2 weeks, IgM is going to rise. Okay? IgA and E also rise in acute infection. However, they tend to fall down even before IgM. Matlab they are very, very quick. Jaldi jaldi rise karo, jaldi jaldi fall karo. That is what is IgA and E. They rise very, very quickly, fall very, very quickly. IgM also rises quickly but falls dheere dheere, acute. They are all indicative of acute infection. IgG on the other hand is indicative of chronic infection. Of course, I'm not telling you anything new in this. This is something you know since your first year MBBS. IgM acute infection, IgG chronic infection. But what is it that I'm going to tell you definitely now is that this IgG is going to be a lifelong wala antibody. This is going to stay for a lifelong. So now you need to know that in early cases kaun sa type ka IgG hota hai, whereas in late cases, because this is going to stay for a lifetime, right, which kind of IgG is there. So in early cases there is going to be a low avidity. The binding is going to be low avidity, whereas in late cases there is going to be a very strong or a high avidity that you will notice for this IgG. So let me show you the application of this. That is going to make much more sense. Have a look at this table over here. Look at this. If I say someone has IgM antibodies and not IgG, you will say IgM is indicative of what? IgM is indicative of acute infection. With that IgM, if IgG comes, don't start immediately calling it chronic, but that IgG, they are saying ये low avidity वाला IgG है. तो low avidity वाला मतलब अभी बस start हुआ है. This is also acute infection. I will call it chronic infection or I will call it a remote infection only when that IgG is of strong avidity. That is when I call it a strong or a chronic or a long-term infection, when I see that IgG of a strong avidity. Whereas when nothing is there, तो मतलब no infection, there is no doubt about it. So simple. When M is there, it is acute infection. Even if IgM and IgG are of low avidity, then also acute. Chronic only when that IgG becomes a very, very strong avidity infection. Now, which out of these, other than that, can you do? Do you have any, like culture, blood agar and all of those, or any other NNN media? No, this is non-cultivable if you are growing it on any of the culture media. You will have to grow it in one of the animals. So some intraperitoneal injection of mice is what you're going to do, and that is how you're going to grow Toxoplasma. I told you, the cycle was also zyadatar animal wala, so animal growth is what you're going to see. Now tell me, despite studying all of this, before I go on to the treatment, what is the gold standard which is still mentioned in the textbooks? The gold standard for toxoplasmosis is gonna be the Sabin-Feldman test. And Sabin-Feldman test is a type of a complement. Go back to your immunology chapter, you will see this is a type of a complement fixation test. This is a type of complement fixation test or CFT. And now I'll tell you what are you complement. Matlab, ma'am, you're going to deal or you're going to play around with the complement pathway of our body. So this is a test in which I get to know antibodies. Ek baat batao, I want to find out antibodies in a patient. What test, what sample should I take? You will say, ma'am, please take the serum sample, because let's assume this is a case of Toxoplasma. Okay? This is a case of Toxoplasma. Let me assume antibodies are there in the patient. Antibody I have got from the patient, antigen I will have to give in my lab. So I give tachyzoids. I have prepared live tachyzoids in my lab and I am going to give. So matlab ye ho gaya, in simple words, I have got an antigen in my lab and my patient has given me antibody. Antigen-antibody reaction hoga. If antigen-antibody reaction will occur, do you know your basics that your complement system will get activated? Of course. So now you will say that if complement system is activated, complement system is always for destruction. Complement system will go and destroy the tachyzoids. Complement system will go and destroy the tachyzoids. That basically means that now if I try to look at the tachyzoids under the microscope, will I see them? No. So I add a stain called methylene blue. And on adding methylene blue, I see that tachyzoids to dikhe hi nahi. I did not see them. Why? Because they have already been destroyed. So tachyzoids have come out to be colorless. So the question that you'll get is that if antibodies were present, the color did not come. That is your starting and ending. If antibodies are there in someone's blood, simple take home message. If antibodies are present, then the color is not going to come in the end. If antibodies would not have been there, then the color, the tachyzoids would have remained as it is and they would have shown the color. So when antibodies were there, the tachyzoids got destroyed. So color nahi aaya. If antibody would not have been there, then the tachyzoids would have remained alive and the color would have come. So ulta chal raha hai. Antibody there, color will not come. Antibody not there, color will come. So question kya aayega? You are doing a Sabin-Feldman test and the color has not come on adding methylene blue. You'll say color nahi aaya, matlab antibody hai. Ulta, color has not come, antibody is there. That is what you understand by Sabin-Feldman test, which is the gold standard complement fixation test for Toxoplasma. What is it about the treatment, guys, that we have to know? I guess everyone is already aware that we are going to give pyrimethamine, and pyrimethamine we always give with folinic acid along with that sulfadiazine. That is the treatment that you have for Toxoplasma, pyrimethamine with sulfadiazine. Done with Toxoplasma, the first organism